very good morning. Today we will learn about the principles of bioprocess engineering. Now from the word, what does bioprocess engineering mean? Bioprocess engineering comprises of two words, bioprocess and engineering. Now, what is bioprocess? Bioprocess is the process of developing useful products with the help of microorganisms or with the help of living creatures, with the help of biological creatures. This is biological process, and engineering we will come to know it further. Now classic example includes curd making. At home we prepare curd. Like we take milk, whole milk, we add certain amount of curd. In scientific term, we will say that we add inoculum. We take milk and we add certain amount of inoculum to it. Inoculum means certain amount of curd, and after some time of incubation what we get is the curd. That is one classic example of bioprocess engineering. And other classic examples include wine making, etc. Now when Alexander Fleming discovered penicillin, what he found was Penicillium notatum was producing penicillin. But what is the logic behind it? That organism, Penicillium notatum, is producing penicillin when specific conditions are provided to it. So it is also an example of bioprocess. Now how we can engineer a bioprocess? Supposingly in an industry, if we have to produce larger amount, bulk quantity of material, then we have to check upon pH, temperature, conditions of bioprocess. So if we can optimize pH and temperature required for the maximum production of penicillin, we can produce it in huge quantity and with economic viability. So that is what the objective of a bioprocess, to produce large amount of quantities required for industrial importance with economic viability. So modern bioprocess technology is based on the same principle, that is combining living matter, either organism or enzymes, with nutrients under the conditions necessary to make the desired end product. Now about the history of bioprocess. In 10,000 to 7,000 BC, first wine making developed. And in 7,000 to 5,000 BC, first beer developed in Egypt. In 5,000 BC, what we love, cheese is developed. And in 4,000 BC, yeast was first used for making bread. And in 5000 BC, vinegar was first developed. And in 500 AD, algae is a microorganism, is cultivated for food production. And in 1600, in the year 1600, fermentation was first used as a term. And in 1680, Leeuwenhoek first observed yeast cell in the beer. And in 1856, father of microbiology, that is Pasteur demonstrated that yeast cell ferment sugar into alcohol and carbon dioxide. And in 1916, Germany produced baker's yeast from acetone and butanol. That means acetone and butanol was used as a substrate. And the year 1923 is the landmark in the bioprocess industry. In this year, one company called Pfizer led to the commercial production of citric acid And statistic shows that in 1989 there is 350,000 tons production of citric acid. So at that time, 1923, Pfizer produced first commercial production of citric acid. Now, bioprocess mainly consists of two things. First is unit process and second is unit operations. When we produce anything in a bioreactor is a vessel in which we produce something. When we produce something in a bioreactor, that is known as fermentation with the help of microorganisms. So this fermentation process is a unit process, and all operations carried out in fermentation process is unit operations. Understood the difference between unit process and unit operations? Fermentation is unit process and all operations carried out on unit process are unit operations. There are many industries which are producing large quantities of bioproducts with the help of microorganisms, and they all use the principles of bioprocess. Now, why bioprocess is used? Why living organisms are used over traditional chemical methods of production? First, they require very low extreme conditions, like they require low temperature, low pH, which is easier to maintain. And second, they use renewable sources of energy which can be renewed, like they are economic and they are eco-friendly. Third, and much of the quantity, like larger production can be there with the help of less energy consumption, means it is cheap process which requires less energy. Now, in bioprocess, either whole microorganisms can be used to produce something, or enzyme extracted from microorganism, enzyme extracted from living thing, enzyme extracted from us, enzyme extracted from microbes, or enzyme extracted from plants is used to convert substrate into product. So, either microorganisms or catalysts, which are biocatalysts, enzymes are used for bioprocess for converting substrate into product to get our desired product. Now, bioprocess has its application in wide variety of fields. In food processing, the example which I have quoted, that is citric acid. In waste management, there are certain reactors, fluidized bed and peg bed reactors, in which wastewater treatment takes place, and it also takes place with the help of microorganisms or enzymes. Third, industries in medical research, like penicillin example. There are many antibiotics which are produced basically by microorganisms. So, bioprocess gives us the medicines. It is very important thing to understand. Fourth, agriculture. Fifth, pharmaceutical development, and many other industry of science. Now, bioprocessing consists of two processes, upstream processes and downstream processes. From media formulation to production of product, it is known as upstream process. That is, fermentation is upstream process. And downstream processes, recovery, isolation, extraction, packaging of product. As I have shown in the slide, upstream processing refers to the culturing of cells and microorganisms to create the bulk of bioproduct. This process is done through culture or fermentation. And downstream process includes isolation, purification and product, getting desired product from biological cells. Now, this is the flow diagram for upstream and downstream processes. First, we prepare media. Media preparation depends on the energy source or nutrients required by the microorganism which is used in bioproduction. It requires carbon source, nitrogen source, magnesium, carbon, nitrogen source as the major energy source, and magnesium, phosphate and sulfur as trace elements. And when animal culture is used, then it requires certain growth factors, certain vitamins and certain heat labile compounds. First, media is prepared to culture microorganism, then it is sterilized. Sterilization is very important process in bioprocess, because if media is not sterilized, if any contaminant will be there, then it will consume our medium and it can outgrow our organism of interest. So sterilization is done by autoclaving at 121 degrees centigrade and 15 psig pressure. And there are certain heat labile compounds which cannot be sterilized using these temperatures, these higher temperatures, because they can degrade. So they are sterilized by filtration. There are other methods of sterilization also, like radiation, using chemical disinfectants, etc. But for media sterilization, we basically use for autoclaving, that is moist heat sterilization, and filtration for heat labile substance. Then we take this media, sterilized media, in a fermenter and we add certain amount of inoculum to it. So inoculum in the sense a minute quantity of culture which with the help of that culture we want to produce that product. So we add that culture and allow that culture to grow by consuming our sterilized media. Microbes grow, and when they are in the sufficient quantity, they are transferred to the production fermenter. They are supplied, microorganisms are supplied with the necessary substrate which is required for product formation. So specific conditions, temperature, pH and oxygen are provided, and with the help of these set conditions and substrate, microorganisms convert substrate into product. So we have our product. Now the challenging task is to extract, isolate and purify that product, because that broth, that medium contains cells plus our product. So if it is intracellular product, then cells have to be ruptured to release intracellular product. And if cells are our product, if cells is our product of interest, then cells are purified from the rest of the broth. Supposingly intracellular product is there. So first cells is ruptured using the techniques of centrifugation, homogenization, and intracellular products can be proteins, basically enzymes. Then we purify it by using the precipitation, two-phase extraction, and further purification will be done by ultrafiltration, chromatography and precipitation. Then finally we have the maximum yield of our product. If all the conditions were optimum, then we get maximum yield of our product. Then we dry our product and then we pack. Now that product is ready for marketing, for supply. And one of the important thing is that at every step quality check is there, like whether it is contaminated, whether our fermentation is contaminated, whether our culture is contaminated, whether our medium is contaminated. So at every step quality check is performed. So from culturing to production of bioproduct, it is upstream process, and from centrifugation to final packing and storage, it is downstream process. So bioprocess consists of two parts, upstream process and downstream process. Now we will recap what we have studied. We have studied what is bioprocess, why it is used over chemical methods of production, what is unit process, what is unit operation, what is upstream process, what is downstream process, why sterilization is required, and what all organism required. What all organism require? Proper pH, proper temperature, proper nutrients, proper oxygen supply. Now, this slide shows some of the bioproducts. Bread we use in every day in our breakfast. It is a bioproduct. Curd is a product which is formed with the help of microorganism. Wine is classic product which is produced by our ancestors in large quantities with the help of microorganism. They didn't even knew that they were carrying fermentation, but these all are the products which we use in our daily lives. Uses the principle of bioprocess. Now, we learn how microorganisms grow in batch culture. That means how microorganisms grow in a fermenter. What are the phases that microorganism faces when it grows? When, supposedly, if we take certain soil sample, soil contains large amount of bacteria, then we have to dilute that soil by carrying out different dilutions. In the last dilution, we plate it on media to get our colonies, to get our microorganism. Now, this colony or microorganism, because previously it was growing on soil, now if we want to grow them on lab, grow them in lab or grow them on our culture or our desired conditions, then it will take some time to adjust. It will take time because everybody needs adjustment, right? Supposedly if I am new, I will take time to adjust here. That is why microorganisms also need adjustment phase. After adjusting to the lab environment, they grow exponentially. So there are 4 phases in microorganism growth kinetics. First one is lag phase, in which microorganism adjusts to the environment. Second one is exponential phase. After adjusting, microorganism grows exponentially. Third one is stationary phase. After a certain point, the growth is leveled off because nutrients are consumed. So growth is not increased. And fourth one is death phase, because all the substrate or all the media is exhausted and then microorganism die. One by one we will see it. Lag phase, this is the first phase of the fermentation process, and the cells have just been injected into a new environment and they need time to adjust accordingly, as I have said. And in this phase, cell growth is minimum. Cells don't grow, they only adjust. They don't grow as in they don't grow in number, they grow in size. Second is exponential phase. In exponential phase, cells have already adjusted. This is the second phase in the fermentation process, and in this rapid growth takes place and maximum growth rate is there in this phase. Okay, cell grow exponentially and very fast in exponential phase. It is also known as log phase. Lag phase, first phase is lag phase, adjustment phase, second phase is log phase, exponential phase, in which cell grows exponentially. Now, at some point of time, cell growth rate will level off and become constant. The most likely cause of leveling off of substrate is substrate limited inhibition. It means that the microbes do not have enough nutrients in multiplying. So they enter a new phase known as stationary phase. Stationary phase is the third phase of microbial growth. In this, the cell growth has leveled off and became constant, and the number of cells multiplying will be equal to number of cells death. No cell number is increasing. Number of cells are constant. Cell death will be equal to cell growth. And one more thing. In stationary phase, many antibiotics are produced. Stationary phase is a home for non-growth associated product, which is due to secondary metabolism. Like when cells are the main product, then we take log phase. But when the product, which is the outcome of secondary metabolism of cell, will be produced in stationary phase. For example, antibiotics, certain amount of vitamins, etc. Now, fourth phase is death phase. In death phase, all the nutrients are consumed and now cells reaches the phase of death. It is the fourth phase, and number of cells dying is greater than number of cells multiplying. The cause of death is usually that the cells have consumed most of the nutrients and there is not enough left for sustainability. They cannot sustain because all the nutrients in the cells are consumed. So, we again recap the microbial growth phase. First phase in microbial growth is lag phase, in which cell adjusts itself. It adjusts to the new environment. Second one is exponential phase, in which cell number increases and they consume all the nutrients. Third one is stationary phase, in which cell growth becomes constant and number of cells forming will be equal to number of cells dying. That is, cell growth is constant. And fourth is death phase, in which cells die because there is no enough nutrients left on which cell can rely on. So, today we have studied Introduction to Bioprinciple Engineering and Microbial Growth Kinetics. In this lecture, the most important thing to remember is what are unit process, what are unit operations, what is upstream process, what is downstream process, what is lag phase, what is log phase, what are antibiotics, what is secondary metabolism, what is stationary phase, and why cells die in death phase. You can refer to any book related to bioprinciple engineering, but Pauline M. Doran, Bioprinciple Engineering, is the good book you can refer to regarding this topic. A very thank you for listening.